Hello, everyone. Welcome in joining this webinar on clinical applications of procalcitonin, PCT, and challenges in measurement standardization. My name is Li Han, and I work in HyTest as the product portfolio manager. Today, I'm very happy to also be your moderator today. In today's agenda, we will have a quick introduction of HyTest and also our product, as well as today's speakers and panelists with us. Then we will have two speeches. Firstly, started with Mr. Thomas Maceto's talk on the clinical application of PCT assays and the current technology. And then secondly, we will follow Dr. Matthias Grimmler's talk on the challenges in PCT standardization. Then we will finalize the webinar with a Q&A session. So please welcome to submit any questions or inquiries that you have during the webinar by entering the comment in the questions field. And by the end of this webinar, if we are not able to address your question during the webinar time, we will be also answer that and then hand out the link afterwards. HyTest was established in 1994, already being one of the most important raw material suppliers for the IVD industry. We are now actively participating in IFCC and AACC, currently ADLM Standardization Committee work. We develop and produce monoclonal antibodies and also antigens as the key components for laboratory tests. We provide a comprehensive product line, including monoclonal and polyclonal antibodies, antigens, plasmas, and serum, covering a wide range of clinical areas, including cardiac markers, infectious diseases, inflammation, neural marker, and tumor markers. And related to today's topic, PCT, we do provide monoclonal and polyclonal antibodies and antigen for PCT assay development. So for any product inquiry, please feel free to also go to our website, hytest.v. And on top of that, we have also various products for other established biomarkers, for example, CRP, IL-6, TNF-alpha. So please feel free to send any inquiries. Before we enter today's introduction for our dear speakers, we would like to launch the first poll question in order to understand our audience needs better. So you will see the question popping up in your screen very soon. Which part of the webinar are you interested in the most? Please select one of the following: clinical application of PCT, or second choice, PCT assay technologies and development. Third choice, to know more about PCT standardization, or I'd like to understand more about PCT in general. We're now seeing very active answers coming in. Thank you, everyone. And you will see the result very soon. Great. Now we can see that the second part, PCT assay technology and development, gets the most vote. Thank you everyone for submitting the poll. And now we're coming back to the speaker part. We are very honored to have our speaker today, Professor Dr. Matthias Grimmler. Dr. Grimmler works as the head of scientific and medical affairs at DiaSys Diagnostic Systems. He also holds two other management positions at DiaServe Laboratories. Additionally, he is also a professor at the University of Applied Science, Fresenius. On top of that, Dr. Grimmler has extensive research experience. He also holds a Ph.D. degree from Max Planck Institute of Biochemistry. It's worth to note that Dr. Grimmler is a member of IFCC working group of PCT standardization work, on top of many other titles in different associations. We are also very honored to have our second speaker of today, Mr. Thomas Masetto, with us. Mr. Masetto works as the head of R&D at DiaSys Diagnostic Systems. Prior to his current role, he has also worked in various positions for more than 15 years in biotech and the clinical industry. Mr. Masetto is very passionate about medical and clinical research. And besides the outstanding working experience, he's also a doctoral student at the University of Düsseldorf. Mr. Masetto is also an active member of IFCC working group of PCT standardization on top of many other titles in different associations. Last but not the least, we're very happy to have Dr. Vladimir Filatov with us as the panelist today. Dr. Filatov works as the group leader of our R&D department in HyTest. He has been working in HyTest for 25 years and have led multiple challenging development projects, which leads to excellent products. For example, PCT, AL6, D-dimer, and troponins. Dr. Filatov is an acknowledged scientist, and his interest of research involve antibody and prototype immunoassay development and production, and many beyond. With that, I'm very happy to welcome our very first speaker on the stage. So, Dr. Masetto, the stage is now yours. Thank you.
Thank you. Thank you very much, Lee, for this nice introduction. Hello, everybody. My name is Thomas Masetto. I am an immunoassay developer and head of R&D immunology for DiaSys Diagnostic Systems. First of all, once again, thank you to our HyTest colleagues for the chance, for the invitation to give this talk today about procalcitonin. The title of my presentation is Standardization of Procalcitonin, the modern Troy. I chose these mythological examples in reference to the difficulties and overall the long-lasting process leading us, hopefully, to the standardization of this parameter. Before talking about procalcitonin, however, we should spend some words about the pathology, the pathological condition associated to its use, i.e. sepsis. Sepsis was redefined in 2016 as the life-threatening organ dysfunction caused by a dysregulated host response to infection. Why is this new definition so important? Well, it introduces three main differences in comparison to the old ones. First of all, now the main role of the infection is immediately underlined. Then it strengthens the difference between sepsis and infection in the dysregulated host response. And finally, now the organ dysfunction is directly mentioned in the definition of sepsis, while it was previously mentioned as severe sepsis. Sepsis is a major concern worldwide, as outlined also by the WHO. It is responsible for approximately 50 million new cases a year worldwide, data from 2017, and for approximately 20% of all global deaths. Moreover, 85% of the new cases occur in low or mid-income countries with significant regional disparities in incidence as well as mortality. And finally, what is more important for us today, its diagnosis is difficult. Indeed, some score systems like the SOFA or the Q-SOFA have been proposed, but they are actually meant more for prognosis of sepsis, not for early diagnosis. For these reasons, a number of possible chemical biomarkers have been proposed also to early diagnose this pathology. And among them, procalcitonin. Procalcitonin is a protein. It's a 116 amino acid long protein with a molecular weight of approximately 13 kilodalton. It is the precursor of the hormone calcitonin, and under normal physiological conditions, it is expressed only in the thyroidal C cells. Consequently, under these normal physiological conditions, its blood concentrations are very low, usually lower than 0.05 nanogram per milliliter. The first mention about procalcitonin as a possible sepsis biomarker was made by Essicott and colleagues in 1993. They reported that in concomitance with bacterial infections or sepsis, the PCT expressions could increase up to 1,000-fold. On the other hand, some viral cytokines, like for example interferon gamma, seemed actually to downregulate PCT expressions. What is important is that under these bacterial infection conditions or septical conditions, the stimulations to produce PCT does not act only on the thyroidal C cells, but on all cells of the body, and this is called constitutive secretion. There are some tissues that appear to be mostly responsible for these constitutive secretions, and they are the liver, pancreas, lung, intestine, and adipocytes. One important advantage of PCT over other possible parameters is its prompt increase after the onset of sepsis or the beginning of the bacterial infections. Usually within 2 to 4 hours, we see already PCT increasing in blood. Moreover, on the other hand, also its half-life appears to be quite rapid, around 24 hours, what supports also its use in the antibiotic stewardship, as we will see later on. As mentioned, the first clinical application of PCT is its ability as a diagnostic marker of sepsis. In this article, Lippi and colleagues report an analysis of the so far most promising or most used biomarkers of sepsis, and we can see that PCT is currently the best one that we have, even though not perfect. We see a quite broad range of specificity, and overall, what is important for the diagnosis, sensitivity, centered on 75% approximately, and 85% respectively. In relation to sepsis diagnosis, the PRORATA clinical trial holds likely the most important role to define the algorithm to start, continue, or discontinue antibiotic therapy based on PCT blood concentrations. As we see in this slide, PCT concentrations lower than 0.5 or even more 0.25 nanogram per milliliter strongly support not to start antibiotic treatment, while PCT blood concentrations higher than 0.5 or even more 1 nanogram per milliliter strongly support to start antibiotic treatment because the patient has or should have a bacterial infection or sepsis. Similar PCT concentrations can be used also to guide the antibiotic therapy after a confirmed diagnosis of sepsis. So once again, PCT blood concentrations lower than 0.5 or 0.25 strongly support to stop antibiotic therapy, and PCT concentration higher than 0.5 and a decrease from the peak lower than 80% suggests to continue antibiotic therapy, while even an increase from the peak suggests to change antibiotic. As already mentioned, in relation of the PRORATA clinical trial, also the duration of the antibiotic treatment can be significantly reduced by using PCT as a guide. Once again, by this article, we see a summary of 6 meta-analyses, all showing significant reductions up to 4.2 days of the antibiotic treatment, antibiotic therapy for the PCT guided group in comparison to the control group. In this context, this further important clinical trial called SAPSA was centered on the antibiotic discontinuations in patients with already presumed bacterial infections. As we can see, the 1-year mortality was significantly lower for the procalcitonin guided group in comparison to the control group, but not only, also other parameters like the 28-day mortalities, the adverse events, the costs of the treatment were all significantly better, significantly lower for the PCT guided group than for the control group. One further proposed clinical application of PCT is its ability as a prognostic marker of mortality, as reported in this study. In multivariate analysis, we see that PCT by far shows the best performances in differentiating among survivors and non-survivors with the odds ratio of approximately 2. Moreover, within the septic group, the median initial PCT value was significantly lower for the survivor group than for the non-survivor group. Procalcitonin then has been proposed also for many other pathologies or clinical applications. Among them, perhaps the most known or the most used one is this lower respiratory tract infections, as demonstrated in this ProHosp trial. Once again, PCT can be used to initiate or discontinue antibiotic treatment in patients with this pathology without significant difference in mortality, ICU admission, or disease-specific complications. And finally, procalcitonin is used also to rule out the source of an infective process, as well as to guide antibiotic treatment in a number of other clinical applications. The take-home message of this slide is that the cutoff used for all these applications is always or almost always comprised between 0.1 and 0.5 nanogram per milliliter. So now, how is PCT measured currently on the market? By immunoassays. There are different technologies as a basis of the different immunoassays, and we are going through them quickly. The thermo original sensitive cryptoimmunoassay is based on this TRACE technology, time resolved amplified cryptate emissions. This technology sees 2 antibodies, one labeled with a cryptate molecule and another one labeled with a fluorophore. The concomitant binding of both antibodies to the same antigen molecule brings these 2 molecules close to each other, and this produces or intensifies the fluorescence signal. On the other hand, the most of the tests for PCT currently on the market are based on the chemiluminescence technology or its derivatives. This technology uses magnetic beads coupled with a so-called capture antibody to block the antigen in the sample. Then we have 1 or more washing steps, and then we have the addition of the second antibody called the tracer, carrying the signaling molecule, and the signal is measured quantitatively. Finally, at the current status, we have only 2 immunoassays for PCT on the market based on the particle enhanced turbidimetric immunoassay technology, PETIA technology. This technology uses a much simpler principle. We have polystyrene particles coupled with mono or polyclonal antibodies, and the presence of the antigen leads to their aggregation, and this aggregation is measured simply photometrically. We don't even need to say this. Each supplier utilizes different antibodies. Here we see just some examples, not for sure all of them. This is the basic structure of the PCT molecule divided in N-terminal, calcitonin, and catacalcin. The thermo original cryptor assay uses 1 polyclonal antibody to the calcitonin moiety and one monoclonal antibodies to the catechalcin. The DiaSys PETIA-based assay uses one polyclonal antibodies to the whole molecule. The Abbott Architect assay uses two monoclonal antibodies, one to calcitonin and one to the position 23 to 101. The DiaSorine liaison uses two monoclonal antibodies as well, one to calcitonin, one to catechalcin. And finally, the two tests by Roche and Siemens both use two monoclonal antibodies, but we don't know to which epitopes they are directed. Moreover, we have to say that not for all these tests, the antigen used to calibrate, so for calibrators and controls, is known. In our experience, we notice that the use of a full-length antigen is preferable to this purpose because this is the PCT form that's expressed by the cells upon bacterial infections or sepsis. To analyze the complexity of these measurement situations, in 2022, we published this article in Clinica Chimica Acta. This article will be also deeper analyzed in the presentation that follows. The article considers and analyzes the 4 external quality assessment schemes, EQA schemes, released by the company RFB and measured in Germany in the whole year 2020. As we can observe from this picture, the 7 most measured tests in this country were compared using the results of the 2 samples of each scheme, A and B. Here we see, as an example, only the scheme number 3. And some clear groups were identified, as we see on the slide. The astonishing point here is that we could notice that some tests measured both the samples twice as much as other tests. Indeed, considering and analyzing the whole results of each test for the whole year 2020, we could define some tentative conversion factors, and why I said tentative, you will learn in the next presentation. We used arbitrarily the thermo test as a reference, and we noticed that the Abbott immunoassay measured as lowest. Then we have the two assays by Roche and Beckman measuring slightly higher than thermo and higher than thermo. And finally, we defined a so-called high recovering group comprising the 3 tests by DiaSys, Siemens, and Biomarker. So in conclusion, we can say that Procalcitonin is at the moment the best biomarkers of sepsis for sepsis diagnosis that we have, but it's not perfect. We have seen a specificity of 75%, and overall, what is important for the diagnosis, a sensitivity of approximately 85%. Procalcitonin can be used also for other clinical applications other than sepsis diagnosis, especially to guide antibiotic therapy for prognosis of sepsis, especially mortality. It can be used in lower respiratory tract infections and in other clinical conditions. We saw that different immunoassay technologies, different antibodies, and likely also different antigens lead to complex measurement situations. But what is very important, the cutoffs used are always the same, comprised between 0.1 and 0.5 approximately for each test. We saw that this heterogeneous situation is depicted also by the EQA schemes. And finally, we can say that in our experience, the standardization, or at least the harmonization of different immunoassays is highly necessary. This was my last slide for today. Thank you all for your attention. I invite you to follow the next presentation by Professor Grimmler, that will come immediately after mine, and I will be happy to take your questions afterwards. Thank you very much.
There you go, perfect. Thank you very much, Thomas, for handing over, and welcome to the second part of the talk today. Thank you also very much, Lee, for a kind introduction, and also thank you very much, HyTest, for giving us the opportunity to talk on this very, very interesting topic of sepsis and PCT. In fact, Thomas already introduced a little bit about PCT and standardization, but please let us for just some moment come back to sepsis. Sepsis and what this means for standardization, because as you can see, sepsis for sure is not a problem that have some isolated countries, really a global health concern we have to face. And especially we can also see that some parts of the world are more prone to this problem. And looking at the second part of the figure here, you see about population and incident of sepsis. What does this mean? Of course, yeah, some countries are doing better in providing suitable diagnosis to really get hands on sepsis. Others are still lacking this good opportunity for really discovering or diagnosis of sepsis. So this means, of course, it's a global problem, and if we talk about assays, we have to consider this. This is a global problem. It's a matter of health. It's a matter of costs. And also, of course, we have to consider different settings, like technical aspects, sex and others, if we want to talk about standardization and which assays to use best. But it's not only related to different countries and the educational state, it is also related to a really upcoming problem we are facing within the last decades, I would say, in a very prominent way. It's bacterial resistance, antibiotic resistance. And here you can see some countries like the Scandinavians are doing very well in caring about antibiotics, less using them. But other countries like some parts of South America, North America as well, and Asia are doing not so good job. The main they are misusing, let's say a little bit in this direction, or too frequently using antibiotics for a too long time. That may, of course, contribute significantly to this kind of resistance. And as we have here, there is urgent need of diagnostic markers that really guide how to use and also how to reduce the time, the duration, and the amount of antibiotics to really reduce this burden of antibiotic resistance. And this is not a minor problem, as you can see here. Around about 5 million people a year are dying from this antimicrobial resistance. And if you consider around about 50 to 60 million people dying a year in total, this is quite substantial. And that's something we have to care. And of course, also we have to find suitable diagnostics and have to really ask ourselves, are today's assay ready for this kind of challenges, bacterial resistant and the global distribution? So coming back also to what Thomas already introduced a little bit, from the right side, the different assay principles you have learned about already. We are really asking ourselves in 2020, how does the market look like in regard to this very well suitable parameter, procalcitonin? Distribution of assays, as you can see here, is around about 87 to the luminescence assay technique, depicted here, around about 15% of the fluorescent assay method, and around about 7% of this particle enhanced method here in Germany in 2020. I think substantially this has not changed so much. Maybe there is a little bit shift towards the blue part, the PETIA assay, but in total it stayed the same since in 2020. So that's the distribution. Let's have again a look how these assays do perform. And indeed, this is from the publication Thomas already briefly introduced some minutes ago, where we really had a focus in the year 2020 exclusively in Germany on RFP ECRIS organization, all the ring trial results performed in this year. So as you can see here in the A part, this is reflecting all the assays, and highlighted in blue, we see the fluorescence method. Coming to the B part, just the same, and of course highlighting the blue part, the luminescence method. And in the C part, we can see this is the PETIA method, less, as we have learned, around about 7%, and easy to really ascribe also to the two different companies that are using the assay. But what you can see is also very clear. It's quite some large distribution of test result we are facing. So coming to the presentation, or the slide Thomas already presented some minutes ago, now down to the manufacturers. A little bit hard to see. So for this, enlarging this a little bit, you see the different cohorts, the different manufacturers form. And the first good thing, it's very clear. There is quite some nice correlation, as you can see here. So manufacturers are doing quite well. But at second glance, you see, of course, this wide distribution. So you would hope that something like this in the clinical setting does not occur, but you are looking for something that is really narrowed down to a small circle, or if you're really better, to a really centered recovery of the controls in a very clear and comparable way. So as you can clearly see, this is not so much the case, at least not in Germany, but probably it's the same in other countries and worldwide. So again, what does it mean? If you look at the two most diverging assays, like in this case indicated here and here, you can clearly see what Thomas already introduced. There is a huge difference in between the two kinds of assays, and of course this has quite some impact into the outcome, the diagnosis by the doctor. I mean, if you consider one of the decision ranges, the cutoff of PCT at 0.5, for example, double, as you can see here, the value makes quite some change in the decision a doctor has to do. So in one case, I mean, patient is healthy. In the second case, it means patient is ill. And of course, this has dramatical impact into the further treatment. So the answer to the question I raised some minutes ago, no, these assays seem not to be in an optimal stage at the moment. So the question is why? Thanks, Thomas. I've stolen some of your slides, of course, as they're very educating. Why this is the case? We have learned there is quite some heterogeneity within the different assays and manufacturers, coming from the antibodies used, where they locate, and also for the enhance and the amplification system they are using, fluorescent, luminescence, PETIA. And in most cases, you only have two antibodies. There is a big exception, as we also heard. This is the PETIA assay, also mimicking a little bit in the crypto method, the reference method. So the big question, the big answer on this is really, yeah, heterogeneity is mostly contributing to the heterogeneity of the assay setup and, of course, of the lack of standardization. So again here, situation of PCT, quite some clear slide. We are measuring with different ways, different rulers, with different outcome in quantitation of PCT. So what to do? And again, the clear answer is yes, we need to have a more detailed look to PCT and how to standardize this parameter really for all the globe. Indeed, this already was not done, but initiated in 2018 by the IFCC, forming an IFCC working group with quite some contributors. So quite happy that so many really people are scientists and participants from companies. As you can see, 16 scientists and 14 companies really participate in trying to understand situation on PCT and get hands-on. And I think this is also a number that is constantly increasing. So many people really think about how to do this work, and some key partners have really joined forces to get more detailed hands-on to provide a reference method. It's a mass spectrometry-based reference method And derived from this mass spectrometry reference method, the aim, the goal of the IFCC, of course, is to get hands-on and provide reference material, a primary calibrator, so every manufacturer has the ability in the joint for really recalibrating a system and try to really get a well-defined ethics based on this reference material and the reference measurement method. So, as I mentioned, under the roof of the IFCC, three big partners, let's say that is, have really joined forces. One is the Laboratoire National de Métrologie, the ESA in Paris. Two other ones are the University of Applied Science here in Germany, near Frankfurt. And the third one is the company DiaSys, who also really joined forces to provide and work on quite substantially since 2018 on really how to quantify PCT by a reference mass spectrometry based method. But what to do and how to standardize PCT? As you can see in the background, if you have some simple material like some coins, also money never is simple, I think we can agree on this as well. But of course, if you have a balance and if you can really touch this material in a very clear way, standardization is easy. It is not if you are talking about a protein, for sure. And PCT, as we have really well heard by the nice talk by Thomas, is a complex protein, that's for sure. It is proteolytically processed. We have seen different fragments on the onset, but maybe also in decline of the sepsis. It is very low abundant, especially in the onset of sepsis. And it may also be post-translationally modified. So for this, you need to know your analyte. And of course, also you need to enrich your analyte if you are talking about standardization of PCT. So please let me guide you a little bit to these two problematic settings. The first one, considering the cutoff of PCT, we are talking about this range 0.05 up to 0.5, giving the really first cutoff. So most patients are in the range of 0.1, 0.2 per milliliter. That means indeed the detection limit you need to achieve by mass spectrometry method is on the limit of what instruments and techniques can really handle nowadays. The second one is you need to know your analyte. So on the left side, you see a little bit here the structure prediction of human PCT is a small molecule, quite well processed, and that have a focus on some special settings. As you can see here, excuse me, special settings on the cysteines here in the background also. They may be prone to oxidation, especially if you remove the analyte from the body, working up, doing your immune assays or also standardization process. Of course, you have to consider oxidation process. You have to work fast. Also, considering that some of these cysteine bridges, may really form a bridge, and of course, independence where the antibody of the immunoassay is located. This may have quite some impact in precipitating or detecting PCT. Indeed, so we had some focus here on this special setting to see, is there an impact? Do we have to care about this setting on PCT? Yes, we do, we need. So if you simply use a recombinant PCT or PCT and let stand for some while, let time for oxidation happen, you see, yes, the recovery of this, let's call it oxidized PCT, is quite low compared to a non-treated. So it's only around about 80% in this case of the CLIA technology, and it's around about 85% in a combination of the polyclonal PETIA technology. So indeed, yes, this is not 100%. So you lose something in binding and detecting. You can convert this a little bit. So what we did here is, in fact, just adding some reducing agents to the buffer or this patient sample and stand for some time, different rising concentration, as you can see here. And you see you can substantially recover immunoreactivity. So yes, PCT is a molecule that can be oxidized. This has quite some impact in the treatment and also the precipitation process. So yes, you need to enrich for your molecule, but you need to really try to get hands-on in quantification of all isoforms, different processing, and of course also all the modifications. It's not an easy task. So how to proceed in this? As I mentioned, the three major partners, LNE, University of Applied Science, and DiaSys, join forces, all being clear we need mass spectrometry methods to get hands on this. Method that is not in the area of immune, but really independent, totally independent of immunoassays. But we were not very clear about how to enrich. So we say, okay, two strategies are well known, so let's try both. The first strategy is a chemical enrichment, precipitation, aggressive precipitation of all the proteins, and to enrich this one later on, digest and analytics. The second one, Fresenius and DiaSys were caring about is immunoenrichment. So really trying to bind the analyte by antibodies, enrich this, and by subsequent mass spectrometry to really give room for analytical and quantification. So just let me try to guide you a little bit about these different complexes, complicated. This is the first strategy that was followed by the LNE in Paris, and very much important. You see, this is not working on its own, so you need internal standardization, you need internal referencing. So what we, or the LNE, were doing both in this case, we need to get hands on highly pure recombinant full-length PCT of really, really good quality. Defined one, and if there are some impurities, have further work in really impurity correction by absolute amino acid analysis. So the first step is really setting traced material, labeled material, full-length material, and this quantified material, this internal calibrator, as to call it, is spiked into a patient sample, as you can see here. Both subsequently are in the next steps precipitated, antibody-free, that means by chemical means, and all the content, the standard, the internal standard, and the proteins is precipitated and digested, and later on it is going in the direction of LC-MS analysis. Of course, there's a lot of activity in calibration of this material and also controlling mathematics and uncertainty evaluation. The outcome of this was giving a summary in this first step. Yes, IFCC, especially LNE, can really provide a validated, robust LC-MS-based method based on detergent precipitation by some detergent acetonitrile, and is reaching the LOQ about 0.25 nanogram per ml. Also covering quite some dynamic range, as you can see here. Of course, possible to extend by dilution of the sample. You need some sample volume, 0.5 milliliter, which, considering this is quite a lot if you're talking about septic patients. And of course, this is needing some time to work up. So it's not an all-day method, for sure not. It is just a method to define reference material and isolated patients to really see how they do, let's say, react and confirm with the immunoassay methods. So that's for the LNE strategy, for the chemical precipitation. Going a little bit more into the second strategy, immunoenrichment. The first question is very clear. Which antibody to use best, as we have heard? Enrichment is necessary, but also you really need to respect to get the most isoforms you can get hands on and also the most modifications you get hands on. So we decided to follow the strategy already presented here, to come back to a polyclonal that in our eyes really can cover all the molecule of PCT we know so far. And for this, we get the step to really bind a polyclonal antibody to some magnetic beads and to work this as a precipitating matrix. So here you can see the workflow very much brief. Again, you have a sample, patient-derived sample. Here again, you have to spike in a labeled PCT full-length material, very well qualified and high quality. Then you add your immunoactivated magnetic particles, some incubation time, of course, precipitation by centrifugation, or in this case by magnetic beads. You do in beads or on beads digest, and the subsequent eluated peptides are analyzed by LC-MS as well. Of course, if you do something like this, it'd be necessary to really have hands on your materials, your tools you are using. So we are using to set up this method per se. In the first step, we used a full-length recombinant material. And you can see here very clear, this is very pure, very good. And one of our first tries were different methods, different antibodies, different settings to have a really titration, how much antibodies, how much particles, which kind of particle you need for really precipitating the majority, all of the PCT molecule of the analyte from a sample. Coming to the second, of course, it's not only the amount very well defined of bead material, antibodies, antigen to get the best ratio. It's of course also reproducibility in the background. So of course this should be stable. It should be possible to really do it twice, 3 times independent from one day to the next. And as you can see here, this is possible by some magnetic beads I have already introduced to you. And also if you can see here, the background is quite beneficial in these magnetic beads. And also the speed, this is possible to perform for the magnetic beads here isolated at different concentration relevant for the medical decision is already over or complete the 100% or nearly 100% within 2 hours. So less oxidation. Also, of course, for sure you never can avoid oxidation at total. There's one part. But of course, also background, the noise is very much important. So in this case, we have a look at different antibodies and precipitation strategies. The red one is a latex-based one. The second one, as you can see here, is a black one. A little bit hard to see because it's quite some low noise, is the magnetic beads. And the third one is the Cephorose, a very common way of precipitating proteins, and both the latex and the Cephorose did not so well chop here. Again, the magnetic beads are really beneficial strategy to pick up this issue. In the second part, okay, we had a look at the specific signal. That means different peptides we were using to qualify and quantify at the different techniques. And here again, we see a quite clear picture of magnetic beads doing the best job, and have a look at the magnitudes here of sensitivity. So these guys here, these magnetic beads, are really able to precipitate by robust means the molecule, but also this is very important. It's quite easy to have a tryptic digest on the beads and to remove the protein in a complete way again from the beads and put it to mass spectrometry analysis. So of course, this is the first setting, and you have to do, as always, if you want to provide a global method, you have to do a lot of tests and validation. So we did, of course, not for one day, but several days. We had a look at different concentrations, all that covering the range at one day, second day, third day, different operators, and to see how they are doing in regard of the CV. The precision and the trueness, as you can see here, is the second part to really consider. And as we have really observed, yes, the immune method is doing quite a good job. But we all agree in working on this. In fact, we were not able, by our 5 to 10% allowance we gave ourselves at a low range, we did not yet manage to go down in a really suitable way. Also, we had a look at the signal to noise ratio and validated this. That means that we had a look at the different peptides, if they by different days, by different operators, to also recover in a quite similar and comparable way. And yes, indeed, we also can confirm they did. So taking this part together, summarizing, as we have heard from the LNE part, yes, there's a validated robust immune method beside this of the LNE, based in our case on the polyclonal antibodies. We have also the same as the chemical precipitation method, LOQ of 0.25 in the really specification we both the LNE and ourselves have given us. We have a little bit a bigger or wider dynamic range. We lost a little bit, let's say like this, and the amount of serum or plasma we need. We have 1 milliliters compared to 0.5 by the chemical precipitation, but also, of course, we need around about 2 days for working up all these different steps I've introduced to you: binding to the beads, precipitating, elution, biotic digest, and analytics. This is something, this is not a routine way at the moment for sure. It's specifically dedicated to qualifying specific sample and reference material. So, ending a little bit this direction, taking home message. So what we have learned, global burden, sepsis indeed it is, considering really what Thomas already told you, 15 millions a year, total deaths, and also 5 millions are associated to resistant. And we heard that PCT really gives us a good hand, a diagnostic hands on differentiation, diagnostic, especially in bacteria. We need really to be clear how to differentiate bacterial from other causes of sepsis like viral or some other guys that causing sepsis. We have seen also, unfortunately, there's a quite variability of methods due to the used antibodies and the system itself among all the assays, and this is causing also quite some differences in the quantification of PCT itself. IFCC have set up a working group in standardization in 2018, very active nowadays as well. Hands-on, getting a reference material and the methods. And we are both, the LNE and ourselves, able to really provide a reference method and probably soon also some reference material. But, and in science always there is a but, not yet where we plan to go. So we are both fixed to an 0.25 nanogram per milliliter threshold for both of the methods. So we still need to improve this method by more sample volume or really getting better analytics to really go down in our goal for, let's say, 0.01, 0.1 to really be in a way that we can get hands on PCT by this method in an easy way. And of course, we have to match and merge both methods also in the next round of testing in between the two methods. So let's see how this will come on. So now it's time for thanking all of you for really having the patience to having an eye and feel on PCT and the work of standardization. So from my side, I want really to thank both the teams, especially the LNE in Paris, for all the cooperation, the University of Applied Science at Fresenius, and also for the DiaSys team. Thanks. And of course, thank you very much also for the audience for really listening to talk and learning and discussing on later on PCT and the stage of standardization design. Thank you very much.
Thank you. Thank you very much, Mr. Maceto and Dr. Grimmler, for the excellent presentation and very educative. Thank you so much. Before we dive into the Q&A session, if I may ask to launch the second poll, which is related to our next webinar topic, that you can see the question already on your screen. If I may ask the audience to answer this poll, please select all that apply. Which other webinar topic would you be interested in hearing next? Cardiac markers, inflammation markers, neuroscience markers, infectious disease markers, or tumor markers, or all of them? All right, we're seeing really interesting result, which will show soon on the screen. Perfect. Cardiac marker. I see it led the poll result. Thank you. Thank you, everyone, for participating. This really helps us in finding the idea for the next one. With that, I would like to welcome on stage our speakers and the panelists to open your camera for the last Q&A session. Thank you. All right, so let's see. We have been receiving really great questions from audience, and I can see from the backstage that the questions are still, the numbers are increasing. So if we're not able to addressing all of the questions during this live session, as mentioned in the beginning, we will share the answers then later in the handout part. But let's see, the very first one. What is the overall status with the PCT standardization? Very great question. And would you, Dr. Grimmler, like to take this one?
I think, yes, it's best from my side. Indeed, this is a very good question. As I mentioned already, we are not really at the end of the story. So at the moment, as we have seen also from the presentation, we have two methods that are doing quite a good job in really picking up sensitivity up or down to 0.25. So one next goal should be to really go down in sensitivity to at least 0.1 nanogram per milliliter with a respective suitable specification or variation. And the second one, we are especially, this is guided and led by the LNE in Paris, have a really try to merge these two tests to really compare these two tests and also to compare the nowadays situation of immunoassays in the world. So we have heard a little bit about the German situation at the moment. There are some publications on the French situation, but as I mentioned in my talk in the beginning of the slides, it's a global problem. So we should really validate and see about immunoassays all over the globe behave, and especially also how our both reference method compare in the setting of this worldwide evaluation trial.
Thank you. Thank you. Excellent. And let's see what's the next. The next question is how to develop a good antibody for diagnosis application. What's the critical technique or methods? Very interesting one. I believe that Dr. Filadov, this might goes to your area then. Would you like to take this one?
Yes, thank you, Lee. Good afternoon, everyone. Actually, first of all, I need to say that development of good diagnostic antibody is not very easy task. Diagnostic antibody, by definition, have to react with a native antigen and particular form of that, which exists in the biological fluid, which needs to be tested. And the diagnostic antibody also have to be resistant to any influence which may affect its interaction with antibody in that biological fluid. Therefore, one needs to know the antigen first, its biochemical and immunochemical properties. Next thing is to mention is that development of antibody with a particular characteristic is not that easy, and it differs in complexity from development of just an antibody. Then you are not aiming at particular properties of the antibody. As for the critical technique or methods, I can say that unfortunately there is no critical technique which guarantees development of good diagnostic antibody. After getting knowledge of antigen, one usually employs several methods for development of antibodies, hoping the broader the method portfolio used, more likely it is select to antibody with desired characteristics. And I want to stress again that main task is to select antibody which reacts with particular form of the native antigen which exist in the biologic fluid which needs to be tested. That's the main problem in this area.
Thank you. Thank you so much for the excellent answer. And I think since the time is running really short, but if we still have the very last question on the list, which is also very interesting, how will PCT continue to be relevant as a standalone test as additional markers are becoming available?
Yeah, I think this is clinical application, so maybe I can take over the answer. Actually, PCT should never, in my experience, should never be taken as a standalone parameter to diagnose sepsis. I mean, we have seen this during my presentation, a sensitivity of 85%. This means that
it's not enough. Yeah,
almost 2 out of 10 patients, septic patients, you don't diagnose. There is an interesting, from my point of view, interesting expert consensus published a couple of years ago by Schutz and other colleagues, reporting that the best way to diagnose sepsis is indeed a mix of probability of infections, clinical picture, clinical condition of the single patient, and measurements of biochemical parameters like, for example, PCT. And I think this should be the way we go to diagnose sepsis.
Thank you. Thank you so much, Mr. Masetto. And actually, we wouldn't have time for any additional to take any additional questions, but we will come back to all of these with the answers together with the handout of today's webinar. And I would like to send my sincere thanks to all the speakers and panelists for your excellent talks and sharing your knowledge with us today about PCT assay development. And also thank you everyone who participates this webinar. I hope you find it useful. And for any inquiries, please feel free to contact us via the information on the slide now showing. So thank you, everyone. Have a great day, and we will close the webinar now. Thank you.
Thank you very much. Bye-bye.
Bye-bye.
Thank you.
Thank you. Bye-bye.